this project, Monmouth working in the coastal lakes, has very deep roots in many years, initially initiated by John Tiedemann. So thank you, John, for your initial work in setting up this framework. But it's really taken off incredibly over the last 2 years with the support of Professor Adolph. And I also want to have a particular shout out to Erin Conlin, who has joined us as a citizen science coordinator. She's a Monmouth graduate. And I think what we really recognized as this project got going, that how much interest there was in the communities and how important it was to engage the communities as the scientists and to be informed by Jason and other professors at Monmouth in terms of methodologies and framework. So really seeing this community grow, getting the communities involved firsthand, really, I think is the really one of our main goals. So you can collect the science, but unless the communities are engaged, unless there's ownership of that science, unless we understand that the actions that we need to take are at the community level, and I see many community partners as well as citizen scientists at the table. I am speaking to you from the shores of Deal Lake, where I live, and so a special shout out to Don Brockle, who has a nice picture behind him of Deal Lake. You can't quite see my house on that picture, Don, but I appreciate you and all the work that you do for the Deal Lake Commission. And I see Laura McBride as well here. And again, a great partnership between the local towns and Laura and the community and the additional folks who are working with Erin take the measurements. So I will stop there and say again how excited I am about the continuation of this work and grateful we are for people taking time out of their busy lives and increasingly hectic and confusing lives these days. Stay focused on things that are important to us in our backyards, and also to learn along the way with each other and build a relationship. So thank you for inviting me, Jason, and thank you everybody for joining us today.
Thank you, Tony. And I also want to acknowledge Tom Harrington's here, who is the Associate Director of UCI and actually wrote this grant initially when we first submitted it. Yeah, so we have a great agenda today. I'm just going to go through real briefly what we're going to cover. Erin is going to give an overview of the program that some of you may be familiar with, but others. So it's a re/introduction of the program. I'm going to go through this amazing data set that has been collected by citizen scientists and try to make some information from the data that's been collected in the program. Since May of 2019, people have been sampling. Vic Peretti is here from the New Jersey DEP, and he's going to go over their latest HAB mapping tool that's designed to be used by citizen scientists. And what I said to Vic was, what a better group to get that in the hands of than the CLONet samplers who spend so much time around these lakes, some of which have real recurring issues with harmful algal blooms. Then Erin's going to talk about very near to her heart as a new citizen science coordinator, the future of CLONet and how we want to keep going and building this program before we open the floor by about 1 o'clock, a little after 1, to discussion from the group. And we have 4 people who have contacted us with things to talk about. Don Brockle from Deal Lake, Faith Teitelbaum from Lake Takanassee, Whale Pond Brook, Gail Rosewater from Wesley Lake, and Laura McBride from Deal Lake and the Deal Lake Watershed Alliance. And of course, there's time for other people to chime in there as well. So I guess without further ado, the last thing I want to say is that I also, if you're thinking this, miss the free Monmouth lunches that usually accompany these events. I've been missing that since about March 2019. But we'll get back to that, and I look forward to the next time we do all meet in person, and we can do that. Okay, so Erin, I'm going to hand the floor over to you.
All right, let me just
overview of CLONet.
Share my screen. All right, so hi everybody, I am Erin. If you don't know me, I'm the Citizen Science Coordinator for CLONet. What I'm going to be going over is just some of the issues that coastal lakes face, and history of coastal lake work that Monmouth University has done, and then just go over CLONet, what our goals are, what sampling with us is like, and how that data is inputted into the computer. All right, so just some problems in coastal lakes. I'm sure most of you are familiar with these. In urbanized areas like Monmouth County, typically all these lakes experience similar problems. In the summer, most of the issues revolve around harmful algal blooms or algal overgrowth. This is when you get that green, blue kind of algae sitting on top. Sometimes it's not harmful. If they're harmful algal blooms, they can produce toxins, which can harm the organisms living in the lakes, and also the animals or other things that use that water body. So these are a problem because they can deplete the water of oxygen or cause harm to the organisms that live there. These coastal lakes typically have a lot of recreational use that might be canceled due to these HABs, which is not good. Another problem is nutrients and eutrophication. So this is when a bunch of nutrients come into the water and that fuels algal growth. So you can end up with a HAB after that. And then again, it'll cause a depletion of oxygen when they start to die and absorb all the oxygen to decompose. Storm runoff is an issue because in the summer times, it can bring heat into the lakes. That'll heat it up, which also fuels algal blooms, or it can bring in more nutrients, which is a problem we see in these lakes. Sediment infillings, it kind of goes in with storm runoff, so it'll bring all these sediments into the lake, and that can decrease the depth in the lakes along with increase turbidity, which is not good because, well, Dr. Adolph will talk about that later, but turbidity is a big issue in these lakes. Microbial pollutions harm the organisms that live in there, and then that can work its way up the food chain, say if you're eating the fish that live in there. And then floatables, just plastics floating on top and things like that, that can be harmful to the species that lives there. So while all these problems in the lakes are similar, the solutions must be made lake to lake because every lake is different in that sense, say like with what's surrounding it. So just some history of coastal lake work. In 2008, there was a coastal lake summit, and at this, they kind of reviewed the issues, some of which I just talked about, and then the issues with lake management efforts. One of the biggest issues is it's a disjointed path that people take. So say we have the lake commissions working on something, but then Monmouth University is working on something else, and then the DEP is working on something else, and they don't talk about what they're doing with each other. So that was a big issue. And they also discussed lakes suffering from same common issues. And then in 2010, there was a publication made, and that was the Future of Coastal Lakes in Monmouth County, which outlined restoration techniques, best management practices, how to improve water quality, habitat quality, overall coastal lake health. And then they also recommended development and implementation of regional coastal lake management strategies. And then in 2012, Superstorm Sandy hit that devastated the East Coast here in New Jersey. And that was a very big issue for lakes because it did a lot of damage to those too. So in 2013, Monmouth University hosted a coastal lake summit to discuss how we could restore ourselves from that damage. Okay, and then fast forward to 2019, that's when CLONet really took off. In the top left picture there, that was the Coastal Lake Summit. So the initiative of CLONet is basically to create a community-based participatory research. And the goal is that, so we give the citizens the tools they need to collect data, and then we take that data and then we give you the knowledge on that. It's also to just open communication. So say a lake, you know more about your lake if you're living around it than Monmouth University could know. So that information being shared is really important. And some of the goals, so we want to establish a water quality database. We've done some really good work this season 2 of CLONet. We have a good database going, and it's important to have that that database over time rather than just say during the summer 1 year, because it's not just about how the water's changing within a certain time, it's about the time frame overall over a long period of time. We want to engage the community, we want to get them involved, and then we want to end that disjointed path. So we want to open that communication up. That's between Monmouth University and the lakes, and also between other lakes as well. So just kind of how CLONet is laid out. So we want to identify common goals between groups. Monmouth University, UCI will act as the central leadership. So we will communicate with lakes, they'll communicate with us, and then we will help initiate discussion between the lakes as well. So just for our sampling kits, we've simplified them since we first began. We used to do nutrient testing. We don't do that with our citizen scientists anymore, just so we can make it as easy as possible for the citizen scientists. So when they volunteer, we supply them with a water quality sampling kit, and we train them how to use it. Dr. Adolph used to do the training with a student, Maria Riley, but now that is my job. So it comes with conductivity and temperature pen, which measures conductivity and temperature. The pH kit, it's pretty simple to use. You just add some drops of the pH indicator, mix up the sample, and then compare it to the range that they give us. And the dissolved oxygen kits, that's what usually takes up the most time, but our citizen scientists really seem to be getting the hang of it pretty quickly. It involves a lot of chemicals and things like that. And then turbidity, we use a Secchi disc to measure that. It's pretty easy to use, and it gives the best overall view of the lake's water quality. Sampling takes about 20 to 30 minutes on site, and that's if you're with one person. Say you go out with a partner, then the time kind of cuts in half. And it's pretty easy to upload the data. We just put it online, the website's right there, and this is what the data sheet looks like. They take some atmospheric measurements along with the measurements I just spoke about. They can write notes, say there was a huge storm, or like the lake was just treated with pesticides or something like that, they can note that. And then if there's a variance in the data, we can kind of link those together. Me as a citizen science coordinator will perform quality assurance of the data. So each week I go through the data and I just make sure that everything seems in order. If there's something that's super far out of place, I can hook up with that person and see what the problem is, go through their data and their equipment. And then data will be publicly available on the CLONet website. It hasn't been updated in a little bit, but I promise you it will be soon. This is just a training session that occurred in 2019 with Dr. Adolph and Maria Riley. These groups usually were about 10 to 15 people per group. Unfortunately, we can't do large groups like that right now. It's a possibility for the future, but not right now. But I think it's beneficial to have that one-on-one aspect of the training, just so we know that the training and testing is being done properly. This is just some highlights of the sampling seasons. This is both of them combined. So for January to March, they sample every 2 weeks. And then April to October, that is when HABs usually start occurring. Phytoplankton is growing, so we want to be sampling weekly for that, because water quality can change very quickly during that time. And then November to December, we go back to every 2 weeks because the temperatures are cooling down and there's not a lot of growing during that time. So going over some of the lakes, Deal Lake has been very good. It's a very large lake, so we need a lot of samplers for that, which is why there's been 9 stations. And we've had 105 samples from them, which is very good. Lake Como has been very consistent with their sampling. We have 100 samples for them since we started this. Lake Takanasi is very good during the growing season. During the winter, it's a little iffy, but they're doing pretty good. Spring Lake, right now, there's nobody sampling. So we want to kind of get that back up and going. But they've had 30 samples, 2 stations for the time that they did sample back last year. Sunset Lake, we didn't have anybody, but now we have Caitlin, who is testing Sunset Lake and Deal Lake. So that's good. Sylvan Lake doesn't have any active samplers right now. but we do have some samples from them from last year and earlier this year. And Wesley Lake is very good with their sampling. Have 110 samples from 2 stations over the past years. And that's it on me. Any questions?
Thanks, Aaron.
I have a question.
Yeah,
I have a
Gail.
question.
Mhm.
Do you need any more, test citizen scientists for Deal Lake? Because my husband and I are doing Brockles Dock, and if you need another person, another section to be tested, we'd be glad to test it on Deal Lake.
We actually have just gotten 3 new samplers, but if that's something you're interested in, we can find a station for you.
Okay. Thank you.
Gail, did you have a question?
Yeah, what about Fletcher Lake?
Yeah, we don't have anyone doing Fletcher. All right, so what Aaron didn't mention is that my lab group of students through Monmouth, we sample all 10 lakes, including Fletcher. But we don't currently have any citizen groups doing Fletcher Lake.
What is a station? Because it says Tackanassie has 5 stations.
Do you want to answer that, Aaron?
I can answer that. Well, we set up stations based on, say the lake will have one on the east side, one on the west side. If we need somebody in the center, we can do that. It depends on the sampler. If they prefer a station, we'll have it there. It's open. And
Yes.
it's just like how many stations that have had data input online. So it could have been once, could have been more times than that.
Most of the lakes we can get away with 1 station at a lake because they're small enough. Deal Lake is incredibly complicated and large for a coastal lake, and we've been working with filling out 5 stations with guidance from Steve Souza and the Deal Lake Commission. And then Faith at Tackanassie, people have sampled, well, you have the 3-lake system going west. So it gets complicated. So each location, we've had different people sampling different locations within that system. Each one is a station. If there aren't any more questions, I see Jack Conover has joined us and was wondering, hi, Jack. Thank you for coming. I was wondering if you wanted to say a word or two.
Well, thank you. I had the wrong Zoom meeting code. I went through Tony's anyway. I found the right one, so I'm only a little late. But yes, Tony wanted me to comment on the Plongere Foundation's commitment to this important work. And we've done other things, obviously, with Monmouth and with the Institute. But I had a meeting with all of you at the school before pandemic, and extremely interesting. And I grew up around here, so it's of personal concern too. And I'll be listening carefully.
Great. Thank you very much, Jack, for joining us. And again, just wanted to acknowledge you in the beginning, and also wanted to acknowledge very explicitly Aaron Conlon, who's joined us as our citizen science coordinator. So with your support, and I think you'll hear that that's really generated a lot of I think, increasing interest and participation from the community. So I will stop talking now and turn it back to the agenda. So I appreciate you joining us, Jack, and everybody else as well.
Thank you, Jack. Thank you, Tony. And thank you, Aaron. I see some questions in the chat. So Aaron Stretz asked, is there any interest in adding chloride test strips during the winter months? Are you thinking about road salts, Aaron?
Yeah, so the New Jersey Watershed Watch Network, we purchased a bunch of chloride test strips, and we're hoping to gather information through the winter during kind of relatively dry periods, and then also after a salting event, after a runoff event, to kind of compare chloride under baseline and then under runoff conditions.
Yeah, I guess.
They're pretty easy to use. They're just little strips. you stick them in the water and it's pretty easy to read it.
Would that be redundant with the conductivity measurements that we're making? Because that's part of the,
I think it would be a good complement to it. I don't know that we have a straight connection between chloride and conductivity, but the conductivity would also be great information to have to add to our study
okay.
anyway.
Okay, thank you. That's a good idea. We'll follow up on that after the meeting.
Okay, cool.
So moving along the agenda now, I'm next. So I'm going to go through an explanation of the data that you all have collected. And give a data update for the end of the season 2. We consider the seasons to end October 31st for no particular reason. Let me get my little pointer thing here. So of course, I have to start by thanking everyone who's provided these data, and point out that that your time, your effort, and your caring is the reason why we have these data, and what makes the program work. And what I'm going to try to do here, what I try to do in general with the data that are submitted through the portal, is return it to you in a way that turns data into information, because numbers on a sheet don't tell us much. Other thing I wanted to use this slide to point out was that what I thought was a remarkable thing happened over this spring and summer, which is that during the COVID shutdown, groups continued to go out and sample. And because of so many laboratory shutdowns, including my own at Monmouth, you were really the only game in town tracking these lakes through that period. And this picture here is a colonial bryozoan, which was sent to us from, I think Faith sent it from, it's from Lake Takanashi, but it's an example of a freshwater organism that likes clean water. In some places, it's considered a clean water indicator. And I'm gonna get my timer going so I don't talk for 40 minutes. So the lakes of concern here are from Takanashi in Long Branch down to Wreck Pond to the south. And one of the special things about coastal lakes, as you all know, is that they're not like inland lakes. They have a direct connection to the ocean in many cases. And sometimes it's only one direction. Sometimes that goes both directions, in and out. And another thing to take note about coastal lakes is how these tend to exist in highly populated, highly urbanized watersheds, which is an important part of their ecology and their biology. These are citizen measurements of conductivity that are made in different lakes. These aren't lined up perfectly, but you can see that mostly it's Lake Como here, in which we see higher levels of conductivity that indicate saltwater intrusion. Deal Lake to a lesser extent. And then the other lakes, it's primarily fresher water without a lot of conductivity. The conductivity of the ocean, by the way, would be upwards of 50,000 microsiemens per centimeter for reference, 50,000 plus. The sites that have been sampled in this very complicated map, so again, from Wreck Pond to the south to Takanashi in yellow, are sites that my lab group, students, and Erin now sample regularly. And then in red are the citizen sites that have been or are being sampled currently. And you can see Deal Lake is kind of a mess of sites. Sunset Lake is snuck in there. There's different stations, Faith at Lake Takanashi, where it's been sampled. Not all of these sites are active currently, but have been at some point. And the other thing I want to point out here is that the sampling is not just a collaboration between Monmouth and citizen scientists, but the New Jersey DEP has contributed a lot to the sampling, both on my students' lab side and the citizen scientist side. So here's the data as of November 10th, and this is a lot to look at. So let me walk you through the different lakes data are shown by different colored symbols, and it's a little fuzzy, but you can read the lakes here. This is all citizen data. Started back in May of 2019 and has continued through till present, and this goes through November 10th. And I want to make 4 points here. One, that the kind of data we're collecting are important, that we have good time series of how the data change over time. That's where we're going to get the information out of these values that we're collecting. Some of the parameters are clearly seasonal, go up and down with the hot and the cold seasons, for instance. And some of the parameters are relatively consistent over the year, like pH. Wesley Lake, for 2 years now, has had these low pHs pretty consistently with a few differences here and there. And then places like Como in the orange, pretty consistently over the year. But you don't see a lot of seasonality to that. So it's a different kind of variability over the year. And then finally, the last point I want to make is that, and Aaron made this already, data that are collected by citizens or anybody in any program have to be looked at with a critical eye. And before they go through the quality assurance, quality control procedures that Aaron was talking about and that we're working on now, we have to consider them as provisional data. And I'll point out this example here. Back over the summer in Como, Elizabeth Ooms and John Buckley were getting some really low oxygen values, and I sent students out to check them, and Aaron's gone out and checked since. And I can't exclude those values, but they are odd compared to the rest of the values. So we have to look at them with a critical eye. I'm going to go through 2 main parameters and try to draw some meaning from them. Those are the Secchi depths that we've been measuring in the lakes, as well as the temperature in the lakes. And there's other parameters. I just don't have time to go into them, but I'm more than happy to talk about them in another capacity. So Secchi depth is what's measured with that black and white disc that Aaron showed, and simply lowered through the water, and we're measuring lake transparency. Deeper depths equate to more transparent water or cleaner water, if you will, less turbid water. And if we summarize all the data that's been collected here in these box and whisker plots, we can see that there are areas like Lake Takanashi that pretty consistently have the deepest average depths. So the clearest water, maybe no coincidence, so that's where the freshwater bryozoa was found. As opposed to places like Sunset Lake in Asbury Park that pretty consistently has the lowest Secchi depths that we've been seeing. Then there's a range in between. There are places like Deal Lake, again, a big complicated lake with lots of different areas where you get a range. The average is low, but there's a wide range of Secchi depths in that lake. And if we look at the time series data here, so we're looking at this second from the top plot, the time series of Secchi depth over time, you can see, let's look at 2020. The red is Deal Lake, and it's pretty consistently low. And Takanashi in the yellow is pretty consistently high, along with Como, pretty consistently high in 2020. And then in purple, Wesley Lake, slightly above Deal Lake. So within a season, pretty consistent. And actually over the year, Secchi depth seems to be pretty consistent in these lakes. There's some, you can see in Wesley, the depths got deeper in the winter here. And maybe we'll see that again this year. So what do we do as limnologists? People who study lakes are called limnologists. What do we do with the Secchi depth? And there's something called the Carlson Trophic State Index. And what Carlson and what limnologists do is basically, the Secchi depth is this kind of global indicator of lake health. If you can measure the Secchi depth and get a good read on that for a lake, you have a good idea about a lot of other properties of the lake and in general, the classification and general health of the lake. These are ranked from oligotrophic, which are the clearest lakes, to eutrophic and hypereutrophic. And the point I want to make here is simply that the coastal lakes, even though Takanashi, for instance, is relatively better than other lakes, in general, we're dealing with hypereutrophic systems. So in terms of pollution and degradation, we're at the bottom of the scale of lakes in general, which means we can only go up. Our Secchi depths tend to be less than 3, more often 2 to 0.5 feet in depth. That's in the hypertrophic region. Chlorophylls vary in the lakes, but we do get a lot of chlorophylls in this value. I didn't show that data. I didn't show total phosphorus data either, but on average, total phosphorus in our lakes starts here and goes up as high as 200 to 300 micrograms per liter, which isn't even on this chart. So we're definitely in the eutrophic and very much the hypereutrophic region. Another, if you look at the characteristics of those types of lakes, it sounds like a lot of our lakes too. Rough fish, carp would be an example of a rough fish, and those are dominant and invasive in our lakes, and part of the problem. We do get discouraged swimming and boating through different events that happen over the summers usually. We get thick algal scums and abundant weeds, so we want to move things that way, ideally. Another thing that Secchi depth can do, if I take the DEP data that's been collected with DEP and my students for total nitrogen and plot it against citizen scientists' Secchi depths, there's this relationship that, again, the higher the Secchi depth, the clearer the water, the lower the total nitrogen in the lake. And that speaks to the other problem with these lakes, an ultimate problem, which is nutrient loading that leads to water quality issues. But also speaks to the value of the data that are being collected by citizens, that essentially with a black and white disk, we can model what the DEP is measuring and supplement those measurements of nutrients. And wow, I am impressed by that. Okay, so Secchi depth is telling us we're in a eutrophic to a hypereutrophic system. Let's talk about temperature now as another parameter. This plot gives the average temperatures by lake, but it reflects very much who samples when, year round versus mostly summer sampling with the higher temperatures. And again, this is a seasonal parameter that changes by season. And I want to draw some meaning from that. And of course, being who I am and what I do, it's going to be about HABs, which are a major water quality issue. We know from physiological and laboratory studies that harmful algal bloom species, the things that cause HABs, like hot water. Specifically, they like water above 25 degrees Celsius. And at those temperatures, they tend to dominate other types of harmless phytoplankton that might grow in those waters. What's really cool about the citizen data on temperature is that we've done a great job of defining the time of the HAB window for the year by taking these time series measurements of temperature. So between about mid-June and early to mid-September, and that's true for both years if you look here, our lakes are most susceptible to harmful algal blooms if the right nutrients come in. This is something that we can keep track of year after year and look at, is this window of HAB opportunity going to expand year to year or contract as temperature changes with climate change? And it's a really interesting parameter that we've captured here. If we look at, these are time series, I know there's a lot going on in this slide, time series of HABs in different lakes, again covering 2019 through the winter and then 2020 summer. Deal Lake, Sunset Lake, big HABs in 2019, they occurred basically through that window. They go away about September. Of course, Sunset Lake did have a bloom that persisted through February, but for every rule, there's a rule breaker, right? And the same in 2020. The blooms tend to occur in mid-June to September. I haven't updated this plot in a while. Now, the other factor in causing HABs in these lakes is nutrients. And we know from studies done collaboratively with NJDEP that the more total nitrogen we have in these lakes, the more harmful algal bloom biomass we get. And this is the average of summer seasonal long sampling that shows this. So there's enough phosphorus in the lakes. We know that because we can measure an increase in phosphorus as the blooms are happening. And the more nitrogen that gets into the lakes over the HAB season, the more HABs we get. And we've shown that in a number of different ways. We also know that stormwater is a major source of N and P to the lakes, especially in these urbanized watersheds. So one of the things we've started to do is look more closely at the relationship between rainfall occurring around these lakes and HABs. And we went back to the 2019 data and plotted it against rainfall, 7-day cumulative rainfall up to the date at which the HABs were measured. And we saw for Deal Lake and for Sunset, during that warm time of year, a spike in rainfall followed by a spike in HABs. And in Sunset, a little quicker response. This is a bit of a lag here. A spike of rainfall followed by a spike in HAB, spike in rainfall, and a spike in HABs. And in 2020, during that right time of the year when the water is warm enough, we see the similar kind of scenario where there's a spike in rainfall followed by a spike in HABs. And we're looking more closely and more rigorously at this statistically as we move forward. And this is work that was done mostly remotely by my students, Carly Nolan and Maria Riley, this past summer at Monmouth University. The 2020 data. So in conclusion, this is what I want to leave you with in terms of the data. We always hear about anthropogenic eutrophication, but on the scale of things, we're actually looking at more of a hypereutrophication of coastal lakes. For better and worse in different lakes, overall, if you look objectively at some of the indicators we're measuring, our lakes are in the hypereutrophic region. As we take actions in the watershed, I hope we all ask ourselves and continue to look for whether or not those actions result in change in the watersheds. Changes in Secchi depth, for instance, would be a great indicator that watershed actions have actually improved water quality. Simple measurement. When you take hypereutrophic conditions plus hot temperatures, you get HABs. And we see that in the lakes that have the highest total nitrogen. Also they have lots of phosphorus in them, presumably coming from the sediments already. The more I think about coastal lakes, I would go out on a limb and say, if you want to understand environmentally what's happening in coastal lakes, start looking at rainfall. And I say that both on the short term in terms of watershed management and stormwater management, and the long term, which I'm not going to go into detail today, but the climate change predictions for New Jersey include increased rainfall and increased large rainfall events. So considering the how coastal lakes seem to be responding to rainfall, I think that's something on the long term we all need to be thinking about. And then the last thing I'll say is that, you know, coastal lakes are special because they're connected to the ocean, and what happens in a coastal lake affects swimming beaches. Lakes themselves aren't certified for swimming, but they do drain into lakes that are certified for primary contact. So we need to understand how HABs, fecal indicator bacteria, and other pollutants in coastal lakes affect these primary contact environments, and keep that in mind as we manage and think about coastal lakes. So with that, I will thank you for your attention and be happy to take any questions. I know I went through a lot quickly, and I will put those slides up on the CLONet website.
Jason, I have a quick question.
Yeah.
Your slide with the pigment measurements, is that a conversion to cell counts, or is that actual RFUs that you had on your graph?
That's a great question. That is fluorescence. Erin has done a lot of work and continues to do the cell counts, and we keep track of the relationship between cell counts and fluorescence.
So on your chart, was it is it in cell count or is
So
it
the chart is fluorescence.
fluorescence? Okay, and what meter
Phycocyanin
are you
fluorescence.
using?
Yeah.
And what meter are you using?
We use Turner's cyanofluor. It's a little orange box, and it's great. It's not as simple as the fluorescence, the handheld meter that I know the DEP is going towards using. We're going to start using that.
Yep.
Also, the advantage of the cyanofluor is it gives you a ratio of chlorophyll to phycocyanin fluorescence.
Thanks.
I just wanted to mention that because 2 summers ago, we had growth in Tackanassie Lake and Princeton Hydro came and raked it. They've been treating Tackanassie Lake and Ross Lake this year. So I don't know how that affects the numbers.
So what have they been treating with?
You know, I don't, there's signs up, but I don't remember what it said.
So I have 2 comments on your question and your comment, Faith. One is that one of the things that I'm going to ask Aaron to do this winter is get that kind of information from all of the groups about what's been treated and what they've been treated with and when and schedules. Because so if it was treated with an algicide, that would reduce chlorophyll and possibly phycocyanin levels, but it wouldn't affect total nitrogen necessarily.
Okay.
I don't see how it would affect, I could see one way in which it could affect total nitrogen, but in general, I would say it wouldn't.
Because our numbers look so good.
Yeah, Tackanassie is consistently clearer water, lower chlorophyll, lower phycocyanin, relatively good compared to other lakes. But then when you hold it up to other coastal lakes, when you hold it up to lakes in general, all of our lakes come out in the eutrophic to hypereutrophic range. I'm looking at the, Aaron asked, so you didn't find a strong correlation between total P and HAB events. So to answer that question, Aaron, if we plot total P or phosphate versus HAB biomass, we do see a weaker correlation than what I showed for TN. But if you look more closely at the data, what you see is as HABs build in the lakes, there's actually this residual pool of excess of inorganic phosphate, which if it was needed by the HAB, it would take it up. That's what algae do. They grow on inorganic phosphate. The fact that it's there in excess and building during the bloom means that it's there in excess. They don't need it. But when we add nitrogen to experiments, and we've done this in the field and in the lab, you know, the more nitrogen you add, the more HAB you get. DEP has made measurements of rainfall induced nitrogen loading that gets quickly taken up. So the short answer is no. I don't think there's a causative link between total in the week to week, month to month sense between total phosphorus and HABs. I think it's more about nitrogen in these lakes. But it's more about nitrogen because there's so much excess phosphorus in the lakes already.
Wait, when you say that, so what if we have these huge houses that have, you know, fertilizers dumped on them all summer and it goes right into the lake? What would that show up as? Phosphorus?
Well, because of the New Jersey laws, it should show up primarily as nitrogen.
Okay.
So if I'm not careful, I'm going to start like lecturing on nutrient cycling and the difference between N and P, and then we'll be here all day, which I would love to do, but they cycle very differently. Phosphorus is going to tend to stay in the lakes and cycle internally, while nitrogen is going to tend to be brought into the lakes and it has a way out. So phosphorus accumulates over the years very well. Nitrogen has a different relationship and can get out. Douglas Haddad, I recall in 2019 there was a dangerous red hab bloom where it was very hot in the summer. Is it dangerous to do testing during these times? My specific concern is the potential respiratory response as I am asthmatic. That's a great question. It could be if you're allergic and have a response, yes. But you could there's PPE that you could wear to protect yourself from that, gloves and a mask.
Hey, Jason, do you mind if I ask a question?
Not at all.
I actually am asking it maybe on behalf, so you can explain to the group. You mentioned something that's really important, and that was the contribution of sediments, of nutrients in the sediments being resuspended. Could you just explain to everyone how that kind of process works?
Well, I'm into Vic's time, so let me ask Vic if,
I'm sorry.
if I can have a minute.
Sure.
Okay. So I don't know if I can explain that in a minute.
Yeah.
Phosphorus, one of the differences between nitrogen and phosphorus in lakes, phosphorus will tend to exist when there's oxygen in the lake, bound to sediments in an unusable form. So the sediments are holding on to the inorganic phosphorus. It's not available for use by things in the water column, including algae. As the lakes get darker and the sediments get less light and they go to what we call reducing conditions and less oxic conditions, the minerals that hold the phosphorus start to release it into the water column, and then it becomes available for microorganisms like HABs to use. And that's, we haven't made direct measurements of this, but what appears to be happening in these lakes is that when a HAB establishes itself, it blocks light from reaching the bottom. That would tend to make the bottoms go anoxic because there's no photosynthesis happening down there. Temperatures increase, and then the sediments start to release phosphorus. That's the best way I can explain the pattern that we see, which is that phosphorus increases over the summer as HABs are growing. Nitrogen goes away because they're using it.
You think, Jason, do you think there could be, Don Broccoli, do you think there could be a direct relationship to some of the carp life we have in Deal Lake and other lakes, that they stir up the sediment?
Well, I think there is, and there are papers that describe that. And actually, we submitted a paper on Deal Lake in which I cited that exact phenomenon. Yeah, carp, when they bottom feed and they stir up the bottom, can release phosphate and sediment into the water column and exacerbate that problem.
Yeah, we might be able to work with you on a test in a smaller section of the lake that's controlled. Maybe we can do that next summer. That's a possibility.
Okay. Okay, I'm going to move the agenda along. Vic Peretti is here from New Jersey DEP, and am I still sharing my screen?
It doesn't look like it.
Okay. Okay, I couldn't get my thing to close.
Okay, so let me.
And talk about the DEP's new citizen science HAB monitoring tool. Vic?
Thanks, Jason. I'm going to share my screen now. Okay. Can everybody see? It's the DEP webpage right now. Okay, great. So I'm Vic Peretti. I'm with the New Jersey DEP Bureau of Freshwater and Biological Monitoring, and our bureau is responsible for monitoring freshwater streams and lakes for water quality and biological community impairments throughout the state, and as well as performing harmful algal bloom response and analysis at our lab. So today I'm going to demonstrate how to use our new DEP algal bloom reporting system and our sampling status dashboard, which both of these could be found on our DEP websites. And these were created in response to the governor's HAB initiative in 2019 to better communicate our HAB data and response. The tool that I'm going to demonstrate is best used using Google Chrome at 100% zoom on a desktop computer. So the easiest way to find the report a HAB tool is to Google NJDEP, and you'll come to our website, which is here. If you scroll down, you'll see harmful algal bloom link. You click that, and that brings you to our main harmful algal bloom webpage. Now, this webpage has a wealth of information, but today I'm just going to go over how to report a HAB and our HAB interactive map. So you can access the HAB link. Just about every page that we have on our website has this report a HAB button. Click on that and you'll come to our report a HAB page. And you could take a tutorial on how to report a HAB. If you already know how, you could go right to the report a HAB, and I'll go through each of these fields for you. So this could be done on your phone. It could be done on your desktop. If you're out in the field, you enter the date you observe the bloom and the location, and you could zoom and put a pin in, wherever you're going here. So, if you're at Deal Lake, you could pin, wherever you see the bloom. So, if you're standing here, we prefer you just put in that exact location so we know exactly where to go when we're responding. Put the name of the water body. Again, if you could please put like the formal name, if it's Deal Lake, put Deal Lake, not just Deal, not just, local names. Try to put the formal name in. The more information, more accurate information, the better we can respond. And there's more fields as you go through it. Where on the water body do you see the bloom, the shoreline, the main body, both, closest street address, municipality, county. And then it gives you some examples of algae blooms. And if you could just click on whatever one or more looks like what you're seeing. And other, if it looks like none of these, you could put other. The size of the bloom, small, localized, widespread, unknown. So if it's something like Deal Lake, you're standing on one shore, you have no idea what's on the other side of the lake, you could put unknown. Bathing beach present. It's a water supply. Most people don't know this, off the top of their head, so you put unknown. Is there public access? Yes, no, unknown. Any person or animal that you know of is sick. Have you seen any wildlife that doesn't look well on the shoreline? You see some dead animals, you could put that information in there. And then any additional comments, including descriptions of the HAB. And for the people on this call, you're doing some monitoring. So, any information you can provide, if you have temperature, pH measurements, you have phycocyanin measurements, you can put that all that information in here. That would just, help us respond to the HAB, when we're responding, what to look for. But, all that information gives us, information to assess, what the triggers may be in the future. So the more data, the better. So anything you can put in this field, we definitely welcome it. And then we ask you upload any pictures. So we would like a close-up at the water surface and a photo of the entire lake, like not a panoramic shot, but just a photo of, looking out into the lake to see if, possibly we could see from our desktop what the extent of the HAB is. And then any other photos that you think may be pertinent, you can put as many as you want in here. And then we just ask for the reporter's name and contact information so we could contact them if we need some more information to clarify some things. Do you represent a government agency? We just want this because, if it's a Monmouth County Health Department, we know we're going to contact them directly and actually coordinate them for response. That helps us in responding. Put all that information in, hit submit, and it's immediately emailed to our staff. Now you could use the, you can still use the DEP hotline. Some of you may be familiar with that, but they only take down basic information. There's a delay in getting us that information. So we really prefer if you use this report a hab tool. Gives us a lot of information, pictures. Sometimes we can make a determination right from our desktop. I mean, if it's obvious, like duckweed, we can make a determination. Okay, we're not going to go out this time. If we get more reports, maybe we'll go out next time. So we use this information to prioritize the response. If there's a likely hab, we'll deploy our staff or partners that we coordinate with to perform a site survey and to take samples. Once our samples are analyzed in the lab, the results and alerts are posted on the map, and I'll demonstrate that now. And you can access that on this link, the HAB interactive map, and there's also a tutorial. Basically, the information I'm going to provide you throughout this demonstration is also there's a video tutorial. So if you want to go back and look at something, you can take this tutorial. It's only a few minutes. Okay, so when you first open the dashboard, you'll see a statewide map of investigated HABs as well as other dashboard features. So shown are sites that have been investigated by DEP and confirmed through our sampling and analysis. The statewide view shows color-coded dots corresponding to the alert levels chart on the right. And you could expand that so you could read that. You could also see the different signs here. You could go through those. These are different signs that may be posted if there's an alert in place. There's additional, I'll go through the map details now. So if you continue onto this map, water bodies may have multiple sites or sites sampled on multiple days. So this statewide view shows the most recent samples and alert levels. In the search bar up here, you could search a water body, a town, and so let's try this out. Go to Asbury Park. And that'll zoom you into the location. Now, once you zoom in, you could use your scroll wheel to zoom in and out. You could click and move, click and drag to different areas. You could use the plus or minus to zoom in and out, or you could use the shift and you could create a box to zoom into a specific area. So once you're at the site you want, you could click on the dot and open a pop-up window with additional information that you could scroll through. So this will have our results. We have our fuel fluorometer results, cell count, the dominant taxa. This is the microcystin toxin information. Well, there'll be pictures at the time of sampling that you could scroll through, what it looked like when we were there. And there'll also be information on HAB beach-related closure. If there's no beach present, an NA will appear in this field. If results recommend a watch or a HAB is not present, a no would be there. If our results recommend an advisory or higher level tier, a yes will be shown, indicating that the beach is closed because of that HAB data. And just to note that we only sample HABs at beaches. Other monitoring like bacteria sampling is required by the New Jersey sanitary regulations. That's performed by the beach facility. So conditions other than HABs that may close a beach, DEP doesn't monitor those or report those conditions. And you can dismiss the pop-up by clicking the X here. So the toolbar also contains a legend. It contains a base map gallery. So you go, if you want an aerial view, you could change the map for whatever you would like. The map extent, this gives you some reoccurring HABs that we've had over the past few years. And then to go to the statewide map, you just hit default map extent, and it'll get you back to the beginning. In the upper right-hand corner of the dashboard, there's a drop down for water bodies. You could scroll through this and pick a water body that we had a HAB, and then you just click all water bodies to get you back to the HAB, the statewide visual. On the left is HAB samples per date. So this is just a list of HABs, sites that we sampled per the date. As you zoom in or out, you'll see that list changing. It corresponds to what's on the map. So the more you zoom in, that list will shrink to kind of what's just on the map. And again, you just hit default map extent, that'll get you back to everything. The HAB alert level overall distribution pie chart is here on the left also. This panel is a graph of all samples that have been taken in the 2020 season, grouped by alert level, and displays by sample count. So this function is grouped by total sample sites and not by water body. So there's, like I said before, there may be multiple sites within a large water body, and each of those may have a different alert level. So if you click each alert level to see the distribution of each alert throughout the state, and you just click anywhere outside here, it'll get you back to the statewide view. So lastly, there's more information on the lower left. There's a link to our website, the advisory guidance, our outreach materials, the dashboard tutorial, our response strategy, again, how to get you back to how to report a suspected HAB. All this information is available on these links. And lastly, the system reflects sample results for suspected or confirmed HAB events reported to us. So there may be other HABs occurring in the state not shown here. We update this every day that we have results. Each day at noon, this map is updated with the most recent results for the water bodies that we've investigated. And that's about it. Like I said, you could take another tutorial to go over this again if you're not familiar with it. But we think it gives you a good view of the data throughout the whole state. It's pretty easy to use once you get into it and start using it.
Yeah, thank you, Vic. It's a great tool, and I think this is a great group to introduce it to who will be around these lakes. Am I correct? And was 2020 not such a bad year HAB-wise in New Jersey?
It was actually, we had more HAB reports and more confirmed HABs than last year, actually.
Oh, really?
Yeah. We spent a lot of time last year doing extensive monitoring at Lake Hopatcong and Greenwood Lake. We didn't do that this year. We went back to our original strategy as a response strategy. So when we get a call, we'll respond. So it's a response program. So you'll notice Deal Lake, we never got a call from about Deal Lake this year, so we didn't go there. So there's no data this year for Deal Lake. So like I said, it's a response program. We get a call, we'll respond. Otherwise, we're not searching the state for HABs necessarily.
Yeah.
If our sampler is out there, we're certainly going to respond. Sylvan Lake was an example. That was one of our Sylvan Lake was one of our routine network lakes. When they were out there, they saw that there may be a HAB present, so we did a HAB sample.
Yeah, there's a question in the chat for you too. Do towns with public ocean beaches have to grab samples, and is there a reporting mechanism for data sent to DEP? Even if it is not published by DEP, is it stored in an accessible way?
So we're putting all of our data on this site. So it's all going to be there. If there's a suspected HAB, if we get a report of a suspected HAB and we see that it's actually not a HAB, it'll come up as a green dot on this map. So all data that we receive and any results we have are going to be up on this map. But if we don't get the data, obviously we can't put it up here.
Thank you. Any other questions?
I want to compliment Vic and the DEP for changing the alert, all those warning signs, because the old warning signs were basically, don't touch your water, you're going to die. So
Yeah,
we're all different. So they really, between Lake Apaca, Deal Lake, and Greenwood Lake, we teamed up and we met with the DEP. I think Vic was at the meeting, right, Vic?
Yeah.
And they responded, and it took them a couple months, but they did a great job. So kudos to you guys.
Yeah, thanks. So I just want to point out we've enhanced our communication. The biggest kind of knock was, okay, you're telling us what we can't do, but what can we do on the lake? So, these new advisories, these new alerts kind of break things down more finely to kind of acknowledge, okay, there's still a HAB here, but it's okay to, go in the water up to this point. So I think we're communicating things a lot better on what you could do as far as recreation. And I appreciate Don. Thanks for the compliment.
Yeah, I'll double that from Don. I think you guys are doing a good job of that.
Thanks.
Well, thank you, Vic. I'm going to move things along again. I don't see other questions. We have Aaron is going to talk about the future of CLONet briefly, and then we're going to open it up for our 5 lake samplers advocates to tell us some news. So, Aaron.
Hello again. Let me share my screen. I'll try to get through as quickly so we have more time for the citizen comments.
Yeah, we're only 13 minutes late. That's a minor miracle.
Yeah. So this presentation is going to be about what's ahead for CLONet for the season 3. The biggest change we have is me in the citizen science coordinator position. So there's my email again. So what I am here for is to make the lives of our citizen scientists easier. I am the one to contact for anything you need, if it's training related, if you need more equipment, or anything like that. So I'm the one who does the trainings. And if you want or need retraining, I can do that as well. Again, no large groups for training right now, but that might change in the future, hopefully. I check the data quality and review the field notes. The benefit of me doing this is I do it the same consistency as you guys sample. So for weeks, April to October, I'll do it every week. And then every other week when we switch to the off season for November to March. And that's good because say you put in your field notes, you need a new battery or something, or you have troubleshooting problems. I can quickly correct those issues for you so you can get back to sampling and getting good data. So other troubleshooting issues I can solve is for the conductivity pens, sometimes they need to be recalibrated. I can provide those standards if you don't have them, or they might need a new battery. I can do that as well. And sometimes equipment just needs to be replaced, so I'll be there for that as well. And sometimes you run out of chemicals in your kits, so I can also replace that. And these kits provide or produce waste. So once you fill up your waste bottle, you email me and I'll bring you a new one and pick up the waste. And the benefit of having me for this is it's a faster collection than previously done. So what I'm focused on is more outreach. We're doing great so far, but there is so much farther we can go. So the capacity for outreach has increased. Some of the things I'm focusing on is improving communication between lakes. One of the benefits of having an in-person meeting, aside from the free lunches, was that you get to interact with these other lake people, and that kind of fuels your passion to keep monitoring these lakes for their water qualities, and just kind of keeps us going. So we kind of want that. Hopefully we'll be able to meet in the not too distant future and get that going again. We have a Facebook page, it's not very active, so if any of you have any other suggestions, I would love to hear them. Collaborations are expanding. We have a collaboration with the DEP, which I'll talk about in a little bit. And then Dr. Sterrett at Monmouth University is considering incorporating some turtle watch into CLONet. So if anybody's interested in that, that'll be hopefully available soon. And then one of the benefits of COVID is that a lot of things are on Zoom now. So I am able to go to commission meetings that maybe I necessarily, like maybe I wouldn't have been able to go to. So we're just trying to find new volunteers, and hopefully we can do that through these commission meetings. And goals for lakes that are new to CLONet. We have 5 lakes sampling right now, so we have 5 more in Monmouth County that we can add. So the first thing you want to do is establish a lake group leader, so the kind of person who advocates for your lake, person who directly contacts me if there's an issue. They can find volunteers. You can look for local residents, people living around the lakes, school groups, they have environmental clubs or things like that. Local summer camps. Summer is the growing season, so that's when we need sampling the most. So if anybody's interested in that, we can work something out. Or scout troops, maybe it'll cover a requirement for a badge. And the goal is to stay connected to me at Monmouth and to other lakes. And for most lakes, you really only need 1 to 3 samplers. Unless it's Deal Lake, then you need like 5. But we are getting new equipment. This is a phycocyanin meter. Phycocyanin is what we use to indicate, is our indicator for cyanobacteria, which is what blooms typically on these lakes. It's being donated by the Watershed Institute and the NJDEP. The one we are getting is going to Caitlin Salduti because she's sampling Deal Lake and Sunset Lake, so we'll get readings from both of the lakes. And it'll give us a better understanding of hab abundance in these lakes. So the collaboration, it is a DEP and EPA collaboration. It's set for December 2020 to October 2021. And the goal is to develop hab risk indices for New Jersey lakes. So through CLONet and Monmouth University, we will lead the coastal lakes aspects of this project. And basically what we're doing is we're implementing new capacities for lake monitoring. So we're going to be doing some cyanotoxin analysis, cyanobacterial genetic analysis, and some handheld hab probes. We hopefully will be getting more. So we can have like 1 per lake. That is the goal. We are in the works of creating a quality assurance project plan. And this basically outlines the whole project, the whole network. So it'll discuss problems with the coastal lakes and why CLONet is necessary, how we train people and the sampling methods for these citizen scientists, data monitoring, data reporting and quality control and data analysis. And the goal of this is to provide leverage when applying for grants. And it is a living document, so it will be constantly edited. And just some potential funding for the future. Just 1 second. So this is the water quality restoration grant project. It's under the DEP and it's a potential source for 2022 and 2024 funds. This money is used to restore, prevent, and mitigate non-point source pollution in water bodies. Right now, the money is in the upper Delaware region. In 2022, it'll be going to Northeast, so we're going to apply for that. But we're going to have the most luck when it's 2024, when they give funding to Atlantic coastal regions. So we have that to look forward to. And then just one last thing, we are trying to make sampling as easy as we possibly can. We've removed the nutrient testing kits because we have DEP running for those. We just want to make it as simple for the citizen scientists as possible. The hab growing season is April to October. So we are trying to use the off-season months to improve CLONet. And what I mean by that is improve the communications we have with each other and fix any issues we came across in season 2 of CLONet. And if you're interested in training or know somebody who is, send me an email. And there it is again. And just another shout out to the Jules L. Plangier Foundation for providing us with the ability to host CLONet. And thank you to all of our citizen scientists.
Thank you, Erin. I don't see any questions in the chat. Does anybody have any questions for Erin? I do see Katie mentioned marine science camps as a source of volunteers. Is that what you mean?
Yeah, definitely. They're all over the state. They're great. We worked as a counselor one summer, and
Okay,
great program.
we'll follow up on that.
I don't have a question, but I just wanted to say hi to everybody. My name is Ed Lippincott. I'm in Belmar. I actually sent my contact information to Erin in the chat. I'm the chair of the Belmar Environmental Commission. We're looking to start up this program in Silver Lake in Belmar. I see a lot of familiar faces and a lot of familiar names from my previous work with the Shark River Cleanup Coalition. So I would look forward to hearing from Erin in the near future and getting this started here. I suppose that the fact that I spoke up, I'm volunteering myself as a lake lake leader or sampling leader. So
Yes, yes, you are, Ed. Thank you.
So I'm sure I'll be seeing you guys all again in the future. And I have several other members from the Belmar Environmental Commission have logged in as well. So we look forward to starting this up in Belmar.
Great. Thank you, Ed.
He makes great honey.
I'm a beekeeper. Caitlin is advocating for my local honey. So
I see a question which I can't answer from Marianne Elas. Is there a similar citizen scientist program for inland lakes? And I don't know. Vic, do you know, or does anybody know?
I think probably Erin. There you go.
Erin, yeah, of course, of course.
Have Erin answer that.
I just have my video off because my cat's going nuts. But it really varies. It's kind of more geographically based rather than specific to inland lakes. So Farmington, I'm not sure where that is in the state. Do you guys, can anyone speak to that real quick?
Is it Farrington?
Farrington?
North Brunswick.
Farmington? Sorry?
North Brunswick, I think, Farrington Lake,
Okay, okay.
the Brunswick.
Yeah.
Oh,
So, I
see.
mean, I
Yeah.
can look, I've been trying to develop basically that to answer that question, an inventory of all of the groups who are doing this kind of community monitoring throughout the state, so that we can track who's collecting what data, and figure out how we can fill in some of the gaps in DEP's data. So I'll look into that and I can get back to you about that for Farrington or Farmington.
Thank you. Where'd my chat go? There it is. Okay. Thank you, Erin. Thank you, everyone. Let's open the floor now. Don Brockle. Don, you still there?
I'm
There you
here.
are. I see you.
Okay.
So it's 1:20 now. We've got 5 speakers, so I know you're about as good at being brief as I am.
I'll try. I'll shoot for it. Aaron, do I get control of the screen? Or? Let me see.
Can you?
I got it. I got it. Okay. Let me first by thanking the CLONet's program is fantastic. You know, I know a lot of the folks that are doing it on Deal Lake, and the results look wonderful. I mean, you know, I was surprised to see a 4-foot depth, so I got to find out what that location is. That was nice to see.
You're showing us
As
all
we
your
know,
email right now, Don.
right. So as we know, a lot of our lakes are filling in, and yeah, we've been doing various different 319 programs. And we did one in 2017 that I'd like to share with you. So let me go to Facebook real quick. So I'm going to start off with, you know, our basic cleanups. We usually do two cleanups for floatables and various other things. So this is one we do on Oxenia every year, at least twice a year, with a catch basin on Hollow Brook, and we do the rest of the lake. This is the old sewer plant, this pumping station on Deal Lake. You see by the Interlake and Circle there, that's getting totally redone, and that pumps the sewage from Winnamassa, Interlake in Lock Arbor, Allenhurst, and it pumps it north about 5 miles to Roosevelt Avenue, Ocean Township Treatment Station. So if you have been going by there and didn't know what was going on, that's what's going on. But it's a big eyesore, and it's going to be a beautiful location soon. Again, we have help from Asbury Park taking debris out of the lake once in a while. And if you notice that little crack in the wall, I'll show you that in a second, how that got repaired. This was the beginning of our flume renovation project. We got $550,000 from the Corps of Engineers to improve the structure so they wouldn't have to expand the jetty for fish migration. They wanted us to blow out the sand once in a while. And to be honest with you, from this effort that's been going on for about 6 years, we learned a lot. I learned a lot. That we have to blow it out early February, March timeframe to open up that channel for fish migration. And I think we have a lot more fish coming in the lake, herrings and various different shads, white perch, and various other fish that are coming in. So that's working out quite well. Let me just go through here. Of course, we have to mask up on the lake, right? So we have a little reminder there. A couple scenes on the lake down in the main end. Marketing Sunset Lake. You guys don't know, but Sunset Lake is connected to Deal Lake right around the high school, the Asbury Park High School football field. A couple other scenes from the lake. Otters. We have 5 river otters on Deal Lake that's been spotted. We got some video from some folks, but I thought that was pretty exciting to mention to you guys. They keep on going up. We got trees. That's the flow. That's a typical flow with a full moon and high tide coming into Deal Lake from the ocean. So just to give you an idea of what we see down there in the ocean. You also see some rust on the building and broken concrete. And so this is the beginning of our restoration project of the building. And it's been, like I said, it's been about a 5-year project going on. And this shows you the inside of the structure. These are new actuators. These are going to be remotely, I could be in Hawaii and open up Deal Lake. That's my plan at least. And some other pictures, that's some of the inside work. If you remember the inside, all new electric, got new electric to the pole underground. And we have cameras that we're going to be sharing with the police, and some of them are going to be public cameras that we're going to put on our website. So it's we've done a lot. And this in there, I'll show you, we improved. You notice all the concrete cracks, and we were just worried about that whole structure collapsing. It was built in 1921, approximately, and with the renovations we got coming on now, and here you go. That's what it's looking like now. So this is the new wall. It's been multiple coats of paint. This was the final coat, and this was a primer coat down here. And you notice the wall, the way it looked in the Asbury side. And I'm encouraged. I think Asbury is even talking about maybe trying to restore that wall. Again, the concrete, the wall painting, renovations, the wall, the cameras. And then on the outside section, I don't know if I have it here. One second. We have a new guillotine gate system. Let me get down here. I didn't go far enough down. We have a 7,500-pound gate that we installed at the surge chamber, and that is right. Where is it? Oh, come on. Here we go. Let's get down here. Here we go. This gate was placed at the surge chamber, down halfway between the ocean and Deal Lake, right where the jetty meets the older section of the pipe system. And this thing, in about a half, there's the other side of the lake. In about 30 minutes, you can lower it by hand with the large wheel. We're going to have hopefully a power drill to do the heavy work, heavy loads. But we're going to be using this during heavy storms, Sandy-like operations where we need to lower the lake, shut down the ocean, and stop the infill of the lake during a major storm. So hopefully Sandy-like storms will be less of an impact upstream. Won't be a total solution, but it's going to help. Again, this is a section of our website here. The Deal Lake Watershed Alliance has been wonderful and getting tours of the lake and doing various different projects on the lake. And I thank them for that. We also, on our most recent project, let me get back to the website here, or the Facebook page. We are now in a 319 grant system, and we are now working on putting in, let me see, I may have to go to another site, 1 second. Okay, that's the old structure. There we go. So we teamed up on a 319 grant with Sunset Lake and Wesley Lake, and we got a $735,000 grant. And the main structures we're putting in are treatment devices. One's going to be on Memorial Drive, basically in front of Galley Pizza, and 2 are going to be on Sunset, as you can see here. These are large structures that are going to clean the water, take the sediment out, the oils out, the floatables out, and send in cleaner water into the lakes. So again, it was a nice teaming effort. It was one we dealt with some of the Senator Beck helped us out with this one when she was senator, and we were able to get the grant. A lot of clean infrastructure stuff we're doing. We're doing floating wetlands, both in Sunset and in Wesley Lake. And we're also going to be doing tree boxes in the lakes. And also we're going to be improving the rain garden in Asbury Park that flows from the train station area to the lake. So there's just some of the things we're doing. And another highlight is we didn't get a grant per se, but NJIT got a grant for treating blue-green algae. And they're going to have I call it the blue-green algae eating machine roaming it around the lake next summer. So we'll see how that works out. But that's it for Deal Lake. And again, thanks to organization, the CLONet folks at Monmouth University for all your work. That's it.
Thanks, Don. So this slide that you're showing right now, when are those infrastructure projects going to be in place? When are they going to be done?
We have awarded the MTDs about a month ago. So within the next 6 months, they'll be installed.
So this is a great opportunity for the folks sampling in those lakes to see if those installments have any impact on the water quality things that we're measuring, like especially the Secchi depth. You're talking about limiting sediment inflow and stuff like that.
That's correct. I believe this takes over 3/4 of the water flow goes through these 2 locations, I believe.
Yeah, that's really exciting, especially Sunset Lake. Sunset's got nowhere to go but up.
Oh yeah, I know.
Thank you, Don.
No problem. Take care, guys.
Faith, I'll let you just ask your question. You're up next anyway to give your report. I see you have a question in the chat, but since you're up, you could just state it out loud.
Okay, so I have my slides here. Not sure if I know how to do the share screen.
Well, you could either email them to me real quick and I'll share them for you, or down at the bottom of your screen, there should be a share screen button.
Yeah, there is.
Okay.
So I need.
Click that and then click on you should see your.
I see you. I see your phone. I don't see my computer. Hopefully. I did send my slides to Carl.
Oh, that's right. I have them, Faith. Let me put them up.
It's just that I'm going to have to tell you when to go to the next, but
that's okay.
Well, the question that I was asking Don is, do you have a report about the number of fish that are coming in now that you have the fish ladder, the way for them to get in? So I would be very interested because we put a fish ladder in at Tallahassee, and I don't know how do we know if it's working?
We don't have a specific count. What's going to be really cool though, we're going to have a camera facing looking at the dam. So at high tides is when the fish come in and out. And I've seen them. I've seen them swim in and swim out.
Really?
But it's a natural type occurrence. There's no fish ladder needed because of the way the dam structure is built.
Oh, okay.
But I'll tell you, the folks in the Littoral Society, I believe, have been doing studies at Wreck Pond, and I don't see a reason why they can't come to your lake and or Deal Lake and do that study also. So maybe
Okay.
some of the DEP folks can help out with that.
All right. Thanks. So, our our Whale Pond Brook watershed comes out at the ocean in Long Branch. So we've been working on trying to keep the water quality and keep it as beautiful as it is now. And in 2014, you can go to the next slide.
So you do see the slides. I wasn't sure.
It's not the whole screen, but. Okay, so we wanted to put some green infrastructure in 2014. We started with ground cloth and some baby plants. Next slide. And this is Ross Lake on the Whale Pond Brook, and this is what it looks like 6 years later.
Wow.
And we got a grant to put in that bench and kiosk. And we have layers of grasses. In 6 years, we have never had any goose poop on this trail because we think it's because we have two rows of tall grasses and the geese are afraid to come up. So next slide. That's another view of the trails. Next. When we started doing water testing, we thought, gee, I wonder if we could do an experiment and look at the other side of the lake. Next slide. And see if we could figure out how to make the water quality better by putting more green infrastructure in on the south side. So we took a ride down a road that is a U shape. It's called Red Oaks Drive. And we looked at the downspouts of most of the houses, and you can see the downspout goes right into the driveway and down into the street. No filtering. So most of the houses on the street look like this. Next slide. And there are no storm drains on the whole U except for one, next slide, right there, which takes all the water that comes down that U shape in everybody's driveways and roofs and shoots it right into Ross Lake without any filtering. So we are working with Rutgers and we're going to put in curb cuts here and we're going to build a bioswale. And then we're going to keep track of when the bioswale gets done. Next slide. And you see these little pink ribbons are little grasses that we've already started growing to see if we could keep the geese off the neighborhood. Next slide. And we're also letting the native vegetation grow along the banks and asking the city not to just mow everything down. And they've been cooperating. Next slide. So this is the next lake that we've been looking at, and this is the growth that we had 2 summers ago. It costs $40,000 for the town to clean up that, and we'd like to avoid it. So we looked around the lake. Next slide. And we see that there's a big hill with very large houses and huge lawns that use a lot of fertilizers and pesticides. And there are no storm drains on this entire road. And it goes back pretty far. This is Elberon Avenue. Next slide. You can see there's 2 storm drains at the corner, but they don't really do anything because the water's just shooting down that hill. Next slide. So there is 1 storm drain at the end of that road, and it goes directly into the lake. There's no filtering or slowing down of the stormwater. So we have, Long Branch has agreed to put into an open space grant a bioswale here. And so as soon as the bioswale is done, we'll record it and see if we can't see if there's some kind of improvement. And so that is how we use the green infrastructure to help the water quality. Next slide. That's just another look of the water shooting down the road. Next slide. And another one of the hill. So one more slide. And I don't know if everyone knows this, but I'm going to put a plug in for the enhanced stormwater rules. Every town has to send to the DEP their new stormwater ordinance. So now is the time to go to your city council meeting and tell them to please use the enhanced stormwater rules. And we think that if we do that, we'll see a difference in our watersheds. So that's what we've been doing. Thank you.
Thank you very much, Faith. Let me check the chat, see if there are any questions.
No.
No question in the chat. Does anyone have any questions for Faith? It's really cool what you're doing, mapping out the storm drains and thinking about stormwater management. You know, 3 years of working on this has convinced me that's really, really important.
Yeah, and that's what the new stormwater rules are all about, is installing green infrastructure.
Yep.
We need
Exciting. It's really exciting to see all these different green infrastructure projects coming into place now with the data set behind us that we have, and the potential to really track changes moving forward.
That's what I'm excited about, so we could publish it. How does it work?
Yeah, yeah. Okay, Gail?
Yep. I had a question. Is there a list of the heads of the lakes and their contact information so that we can independently of you sometimes contact each other?
I think we have that within our records, Erin, don't we? More or less.
Yeah, we have it.
And that's exactly the kind of information that we're supposed to be getting out to all of the lake advocates. So we'll put together a formal list.
Okay.
And distribute it. We'll put it on the website.
Great. I mean, I know some of the people here aren't kind of the contact person for their particular lake. So I'm most interested in that, because part of when I talk about the geese, part of the issue, I think, was that Deal Lake and Sunset Lake had some a process in place to deal with their geese. And we can't be sure, but we noticed a big increase in the number of geese around Wesley Lake this spring and summer. And that was kind of one of the things that motivated us to really, do something about it. And I also wanted to say that, I have learned so much from Don Brockle, because I don't really know anything about lakes much. And with what we could all learn from each other would just be amazing. So I hope that the people are open to talking. I know people don't want to do more meetings, so I understand that. And with the 319H grant, with the 3 lakes working together, that has also been, so thank you, Don, for that too. That has been an incredible process. And the whole committee that's in charge of that 319H grant has been so receptive to the Wesley Lake Commission, especially Doug McQueen, working with them on designing what would be best for Wesley Lake. And that grant is all about water quality and stormwater management. And I also wanted to mention that the planning board in Asbury Park has taken on stormwater management as one of their major concerns, especially their attorney really interrogates the developers as to how they plan to manage the stormwater that will come off of their new development. And you know, Asbury Park is full of being developed right now. So we really have a very strong supporter in working with the Wesley Lake Commission in terms of the planning board. It's really been quite amazing to listen to some of those planning board meetings. So the partnerships that we all have, I guess my point is just so important in doing anything with any of our lakes, because we all affect each other. And Wesley Lake doesn't want to chase all of our geese just over to the next lake. And I've been trying to contact Fletcher Lake, which is why I asked you, Jason, and Fletcher Lake hasn't gotten back to me. They may not even have a contact person at this point, but I'm afraid our geese are just going to all go there. So with this influx of geese that we had this spring and summer, our commission members, especially Keith Fiore, who lives on the lake in Ocean Grove, said, you know, we've got to do something, and we've got to do a number of things at once. So we have implemented a multi-pronged approach, 4 steps to try to control the geese. And the first one was what Keith investigated himself, and it's installing solar lights down the middle of our lake. And two of our commission members, Keith and Ed, installed these lights by dropping them into the lake, which wasn't that far down, evidently, with cement bricks attached to them. And there's a string of 10 lights down the middle of our lake now that are completely solar powered, and you can't even see them in the daylight. But at night, I can actually see them from my house, as I'm sure Doug can. And for the 10 lights, it was about $3,800, and the commission did pay for that. And we got them from a company called Away with Geese, which is in Ohio. And we have noticed a slight decrease in the number of geese around the lake. We can't tell you if it's a result of that or not, but you have to install them in August, because you can't, if you do it any earlier in June or July, the geese are molting and they can't fly. You probably all know that. So you have to wait until they're finished molting, and August was a perfect time to do it. Oh, that's beautiful. Yes, that's our lights. And Neptune provided a boat, and they provided a employee to run the boat, and Keith stood on shore, and Ed dropped them along the middle of the lake as Keith pointed to him, you know, where to put them. So that was the first thing that we did to deal with the goose problem. Then we talked to the city of Asbury Park and asked them if we could be included in the contract they had for Sunset Lake for the dog chaser and the egg addler. So they spoke to Neptune. We had a Zoom call about this issue, and Neptune and Asbury will each pay for half of it, which is how the Wesley Lake initiatives work. So it's about $8,000 per town for the year for them to fund the dog chaser and the egg addler, plus $85 per nest if they have to deal with a nest. And that's done March through May. You do need a permit for that in case you're not familiar with it. The name of the company that's doing the dog chaser egg addler is called Geese Chasers LLS, and the city has used them before with Sunset Lake. The next thing we decided to do was educational signs. I know a lot of the lakes have the educational signs around them. And again, this was a partnership with Deal Lake. Deal Lake made the contact of the vendor, and Keith Fiore, one of our commission members, designed them. He's a graphic designer, and I'm not very good at sharing screens, so I'm going to do it the old-fashioned way. I don't know if you can see this. designed a sign. It's a beautiful sign, and they're going to be hopefully on Deal Lake and Sunset, educating the public on why it's bad to feed the geese. And anybody who would like a better copy, I can email you the sign, and Don also has them because Don's getting them actually done after Keith designed them. The last thing that we're going to do in February and early March, and it's called applying Rejexit, which again I learned about from Don Brockle. It's a great flavored food additive that makes the geese not want to eat the grass because it's distasteful for them. It doesn't hurt them. It is approved by, the government to be non-toxic, non-harmful. And Deal Lake also had success with it around their boat ramp. So that's about $2,500 to do completely around Wesley Lake. And we're using Cowley Pest Services, and the commission's going to pay for that too. So some of the things that the cities, the municipalities are paying for, some of the things the commissions are paying for, and hopefully by next summer, we will see that we have a difference in the number of geese around the lake, because it really has been quite significant this year. Thank you.
Thank you, Gail. So from the, what was the biggest nuisance of the geese? Was it their physical presence? I know they can be aggressive sometimes. Or were people more concerned about the nutrients and the goose waste?
Well, I'm gonna, Doug's still on. He may have an answer to that also, but I think a lot of it was the amount of goose poop around the lake. And the lake is supposed to be used a recreational usage. And I take a walk around the lake every day. It is hard to walk around the lake because there is so much goose poop. It is absolutely amazing. Beautiful. That's it.
Nice. Very nice.
So I would say
come to Ross Lake, and we'll show you how the geese don't come up into our garden. It's like, I don't know why, but all we did was plant those rows of tall grasses. So
we have, yes, I agree completely with that. Our Environmental Shade Tree Commission, and Doug has worked a lot on this also, have put in a lot of gardens and plants around the lake. It is getting pretty full. But the issue that I've heard is that geese like to be able to see. And so if there's plants around that are kind of hiding their view, they'll stay away. And so that is also another fantastic way to control the geese is to keep the planting. And we have been doing that.
Okay.
We've had about 6 or 7 cases, physical cases where people walk in front of me and are feeding them with bread and grain and you name it, they're feeding it, and they're walking out of their cars or walking across the house, dropping them over the wall, all sections of the lake, not just on the main section. So
yeah, the signs are important.
Yeah, I mean, look, there's signs up now, don't feed the fowl, you're gonna get fined. We figured maybe a little education, and Keith worked up some wonderful artwork, and we worked on the wording. I think we got a wonderful, I got them in my car, believe it or not, and we're going to start putting them up. So you'll see them pop up along the shoreline. We're also going to bolt them to the bulkheads.
Can you share them with everyone else?
I'm sure we can share the design. Sure.
Okay, that would be good.
Yep.
Well, thank you, Gail. Okay, Laura, last but certainly not least, Laura McBride is going to tell us about our rain garden project on Deal Lake. You need to unmute.
Hi, how are you?
Good, how are you?
The Deal Lake Watershed Alliance works in collaboration with the Clean Ocean Action, other watershed associations, as with Faith, with the Deal Lake Commission. And our most recent project was to build a rain garden on a lakeside location of Deal Lake, off Wickapecka Drive. And I will screen share. Okay, so this is the garden. This is Deal Lake. The nice reason for doing it here, in addition to the benefits of a rain garden, which captures stormwater runoff and keeps it from ending up in the lake, is also that this particular location has access to Deal Lake. So our association is trying to encourage usage of the lake so that we end up with more people feeling as if they're stakeholders in the care of the lake and improvement of the water quality in the lake. This project involved, oops, what did I do? All right, hold on, there we go. So in order for the stormwater, which rolls down Wickapeca Drive, in order for it to be captured and filtered over to the main rain garden, we had to do a curb cut. There's a flow pad here that was designed to stop the water and direct it through the stones, through this rain garden, through a trench drain over to the rain garden that you saw. Oops, again, sorry. This is the main rain garden, and again, the flow pad trench drain over to this garden. So our plan is to continue beautifying this lakeside park. Ocean Township gave us permission to do the garden here. We had a couple really successful fundraisers. So we used some of the funds that we raised to offset the cost of this. Rutgers, who we worked with to build the garden, designed the plans, picked up the cost of all the plants, as well as part of the cost of the curb cut and trench drain, which was really, very helpful, because this part of the project was the most expensive part, that is getting the curb cut and the trench drain put in. We also had donations from a local mulch. Mickey Benoit donated all the mulch we needed for the gardens. The township donated all of the compost. They're also seeding the park area. Our future plans will include having benches so that people can sit and see the beautiful view of the lake. And then additionally, we're going to try to improve the areas where there's lake access. So the Deal Lake Commission, with Don Brockle's guidance, built a kayak rack or boat rack down by the lakeside, but it's so overgrown and really inaccessible at this point that we're going to end up using the guidance of a local plant person to make sure that when we clear that area, we not only keep some of the natives, but get rid of the invasives. And so that park area stays more beautiful. Our goal is to do rain gardens throughout the watershed. As Faith mentioned, it is really important to look carefully at where we can improve with green infrastructure to keep stormwater runoff from hitting our lakes. And as Jason mentioned earlier, and you can just by his slides alone, you can see the correlation between rain and HABs. And if we don't focus on capturing stormwater runoff, we're going to continue to have more harmful algae blooms. So we're going to just keep doing the work that we do. I also agree with Faith. It's really important for us to advocate for enhanced stormwater ordinances and regulations. And I really believe in advocating for the stormwater utility. If we can get townships to adopt a stormwater utility, we're going to have funds going forward to help do projects like this and more. Okay, and that's all I have.
I just want to say, Laura, that Tom Harrington said that he would help us get the towns together to talk about a stormwater utility on a regional basis.
Awesome.
So, yeah, that's awesome.
I think that is the most exciting topic to talk about and to encourage people to not fear, because I think the spin is that it's another tax, and we all know taxes are high in New Jersey, but it's not a tax, it's a fee, and you can reduce your fee by including green infrastructure on your own property.
Yeah.
So it's a win-win.
But the thing to do now is to just get those enhanced stormwater rules passed without the stormwater utility in it.
You're right.
People don't know what it is yet. And
You're
then next time we'll do that when we have a big plan and work together. But I think those enhanced stormwater rules will just make such a difference on the watersheds.
Yeah. And I mean, even using some of what Jason, some of his slides, if it gets to the point where we have the floor at a council meeting and we could just simply show the correlation between harmful algae blooms and stormwater rain events, you know, you know, it's very telling. I found that the most interesting takeaway from this meeting, you know, because it's just so clear cut.
Yeah, and I think that relationship, I've worked on rainfall phytoplankton relationships for a long time. And I think in these lakes, it's especially tight, the connection, because townships tend to funnel stormwater with nothing in between it and the street straight into these lakes.
Absolutely.
So I'd be more than happy to present some of the results of the CLONet group and my students on rainfall HAB relationships. You know where to reach me.
Yeah, I do.
Okay, thank you very much.
I haven't been in touch in a while.
Yeah, well, everything's been weird for the last, I don't know how many months it's felt like.
Yeah.
Maybe in January, we can get the towns together and do some education and use your slides and the pictures we have of what we've been doing on the watershed and some results of testing, and just educate these people, because once they know, how can they think that it's not the correct answer?
You're so right. I totally agree. And January is kind of a slow time. So
that is the time. We have January and February to, you know, we're not up to our necks in HABs yet.
Right.
That's the time to do it. It's a great idea, Faith.
I agree.
Thank you. We'll tell Tom.
He's here somewhere.
Okay.
Okay, we are just past 2 o'clock. I want to thank you all for coming. I'm really excited to have such a turnout and to hear the updates and the interest in the program. Thank you, Vic Peretti, for presenting about the HAB tool. And thank you everyone else for your input, and we'll be in touch. We may have another meeting in March, maybe before the season gets pumping. And until then, unless there's any other questions, let me check the chat. Faith, you have a fan in the chat. I don't know if you've seen that. Awesome. Okay. Thanks, everybody. Have a great rest of the day.
Thanks, Jason.
See you guys.
Thank you.
Good seeing you, Jack.
Yeah, thanks for staying, Jack.
No, I love this stuff. I have to educate myself. I grew up right across the street in Colonial Terrace from where that new rain garden, excuse me, is.
Oh,
But I mean, I was there every day, and I know exactly that part of the lake is quite full of silt, and we're losing it.
Oh, yeah.
yeah, the Colonial Terrace section is definitely silting in. It's almost terrestrial in some parts.
It is. We're going to walk on it.
Yeah.
There you go.
Thank you.
All
Thank
right,
you.
see you later, guys.